Well, thank you. I really want to thank Vivian for the opportunity to talk here at Brookhaven. I've been here many times to collect data, but it's really exciting to be here. It's also amazing to lecture in this room. I often teach Gen Chem, and I have 300 students in an auditorium, but I have never lectured in a room with a balcony, so this is pretty cool for me. It's also really fun because I'm giving a slightly shorter scientific talk than normal because at the end of my talk, I'll talk about some mentoring work that we've done at UMass. But this is essentially the talk that I give wherever I go in the world to give a talk about our work. And the two most exciting stories are both crystallographic stories that are derived from structures that we solved here at Vivian's beamline X6A. So it's really fun to be able to bring that story back to where, where the work really began. So I'm going to talk to you about these proteases called caspases. And caspases are important because they're involved in, as Vivian mentioned, in apoptosis, which is cell suicide. And although that sounds rather graphic, it's really something that happens all the time. So these are healthy cells that we grew in culture. And if we treat them with a drug, staurosporin, that makes them undergo apoptosis, then you can see that they have these gross morphological changes. So they go from looking like elongated healthy cells to be rounded up, and they've blebbed their membranes, and they're committing suicide. And this is not always bad. In fact, in each of us, 50 to 70 billion cells die every day due to apoptosis. And it's really important that they do. Because when apoptosis doesn't happen properly, it leads to disease. So John Reed, who's probably the most famous apoptosis researcher in the world, at the Burnham Institute in San Diego, says that 50% of diseases with no suitable treatment are the results of irregularities in apoptosis. So as a consequence, there's lots of interest in being able to regulate apoptosis. In diseases like stroke, heart attack, and Alzheimer's disease, there's too much apoptosis going on, and your heart or your brain is dying. In diseases like cancer and autoimmune disease, the problem cells are able to persist because they've actually turned apoptosis down. As a consequence, there's great interest both in making apoptosis inhibitors for diseases like this and apoptosis activators for diseases like this. So apoptosis is a field that has thousands of researchers. If you search for caspases, which are key players in apoptosis, you will find over 50,000 references, which means that apoptosis is much more complicated than this slide shows. But what I want to focus your attention on is that there are both upstream caspases, caspase 9 and caspase 8. Those are called the initiator caspases. And they activate these executioners. I have a student who's from New Jersey. So Linda, you said you're from New Jersey. Well, she uses hitmen as an analogy, and she says that these executioners are the hitmen of apoptosis because what they do is they're proteases, which means that they cut other proteins. They're these key proteins in the cell that help a cell survive, and if these executioners cut them, that leads to cell death. So regardless of all the complexities of apoptosis, they all converge on these caspases 3, 6, and 7. So that's where I'm going to focus my talk today, on these hitmen of apoptosis, the executioners. So we were already working on these, and then in 2006, 2 papers came out that made us even more excited about one of them, caspase 6. And one of the studies was this one from Galvan. They were interested in an Alzheimer's mouse model. And there was some indication that if caspase 6 cleaved Alzheimer precursor protein, then it had an impact on Alzheimer's disease. And so they did this experiment where they took mice that have the normal cleavage site. So this Alzheimer's precursor protein can be cut by caspase 6. And those mice get synaptotoxicity, just like in Alzheimer's disease. But if you take this site, which is normally where caspase 6 cuts Alzheimer precursor protein, and you change one of the amino acids from an aspartic acid to an alanine, now caspase 6 can't cut that site. And those mice are protected and don't get Alzheimer's disease. So this suggested that if we could inhibit caspase 6, we could treat Alzheimer's disease. That same year, Michael Hayden's group at UBC found a similar effect in Huntington disease. And he's found that if you took polyglutamine expanded Huntington protein, which is what causes Huntington disease, and you make it so it can't be cleaved by caspase 6, those mice are protected from Huntington-like symptoms. So in 1 year, it became evident that caspase 6 cleavage was a common theme in multiple neurodegenerative diseases, and that made caspase 6 a target for drug development. But you'll remember from the previous slide that caspase 6 sort of works in a similar pathway with caspase 3 and caspase 7. And if you block all those proteins, caspase 3, 6, and 7, you're going to give your patients cancer. So if you want to use caspase 6 as a drug target, you need to specifically inactivate caspase 6, but not all the rest of the caspases. So that's where we got really interested, because we're interested in something that allows us to achieve specific inhibition. So before I get to that, I want to teach you just a couple things about caspases. Caspases have this funny name. If you're a biochemist at all, you are wondering, where does this come from? It doesn't tell me much about its mechanism. Well, it gets its name because they have cysteines at the active site. They cleave their target proteins right after aspartic acids, and they're proteases. So somebody came up with this clever name, caspases. And they're dimers. So the active conformation of them has 2 chains of the polypeptide coming together. And for the crystallographers in the audience, this is a really unflattering view of our structure. But here's a nice view that is a genuine crystal structure. And you can see that this protein is composed of 4 chains. 2 large subunits, which are the dark colors, and 2 small subunits, which are the small colors. And what's important is, this is where it binds substrate. In order for that to bind substrate, you have to have something happen. These loops, which are generally disordered, have to talk to each other. So this L2 from the red subunit needs to talk to this L2 prime from this light blue subunit. And if those 2 don't talk to each other, the enzyme isn't active. So I want to show you a little bit more about that. So here's a structure of caspase 7. This is work that I did when I was a postdoc. Caspase 7, this is the dimer. Here's the 2 active sites. And here's a cavity in the middle of the enzyme. And what we found was that small molecules like this can bind inside, can bind inside this cavity and, just take a little drink here, and inactivate the enzyme. And based on a bunch of crystallography, we can understand how that happens. And that's what this movie shows here. So in the zymogen, you see that these loops are disordered. When the enzyme binds substrate, I'm going to play that movie again because I think my tongue is tied today. So in the zymogen, these colored loops are disordered. When substrate is bound, then this purple loop, which is L2, interacts with this green loop, L2 prime. This residue gets pushed down, and this residue gets pushed down. You guys are going to know this movie by heart by the time I get through it. So there's substrate binding. And now, when this small molecule binds here in this cavity, then 2 things happen. This green loop gets locked in a down position, tyrosine gets pushed up, and arginine gets pushed up. And so we can really understand a couple things about this enzyme. One is that in order for this enzyme to be functional, these loops have to be in the right conformation. Another thing is that this enzyme is super flexible. And so we already knew that this small molecule worked by an allosteric mechanism. So some of you, I know, are not scientists, so I want to teach you a little bit about allosteric mechanisms. So I'll call this slide Allosteric Sites Explained. So let's imagine that our drug discovery goal is that we want to insert medicine into the appropriate orifice. That would be the active site. So I have here a model of a little protein. In this case, it's a baby. And here's its active site. So if we want to insert medicine into this active site, we need to get the mouth open, right? So we could either try to force the mouth open. That would be active site inhibition, right? If we just force the mouth open and put the medicine in. Or we could use an allosteric mechanism. So here you can see an allosteric mechanism where my husband is tickling the foot of this baby, and when he tickles the foot, or the allosteric site, then the mouth on the baby spontaneously opens. So that's just like an allosteric, that's just like allosteric sites in proteins. So every baby's mouth or active site is pretty similar, right? They all don't have teeth, they have a tongue, they like to suck on bottles containing milk, but their allosteric sites are unique. So some are ticklish on the foot, some are ticklish on the knee, some are ticklish on the hand. So if you wanted to make a selective inhibitor for each baby, you could make an allosteric inhibitor that only tickled their far away from the active site, their foot or their knee. And so proteins and babies are pretty similar in that regard, that they have these unique allosteric sites. So we want to be able to specifically inhibit caspase 6, but no other caspase, because we want to be able to impact neurodegeneration. So we think allosteric sites are the way to go. And so Edwin knows Ellie Velasquez. We'll hear more about him today. But Ellie said, well, I wonder if those two compounds that can inhibit caspase 3 and 7 can also inhibit caspase 6. And indeed, he found they could. So then we knew that caspase 6 had at least one allosteric site, which was at the dimer interface. But it turns out that this allosteric site is the exact same site that's present in caspase 3 and 7. So is this going to be a good site for targeting caspase 6 specifically? No, Linda knows. No, this is terrible, right? It's going to hit 3, 6, and 7, and you're going to give your patient, while you cure them from Alzheimer's disease, you're going to give them cancer. So we weren't, unfortunately, this site was not going to work out for us. So that's when Shravanti Vaidya started working on the project, and she worked for a long time to get crystals of caspase 6. And when she finally did, we brought them here to X6A, and she collected data. And then she got this electron density map. And when she saw this electron density map, she was really perplexed, because caspases don't really look like this in this region. But our electron density map was very clear. We had a helix, a strand, and another helix. So when she finished refining her structure, she found that caspase 6, which is the orange one here, looks very different from all other caspases. So in green, we have caspase 7. And what's different, so what we've done is we've superimposed those here, and then we've zoomed in. And if you look at caspase 6, the orange one, you can see that it's got this long helix, whereas every other caspase has these 3 strands and a short helix. This helix is also extended, and this helix rotates out. And Shravanti was very fascinated by this. She said, wow, that doesn't look like any other caspase. It's very different. But I wonder if this can actually bind substrate, because this extended helix pokes right up into this pocket where the substrate needs to bind. So, of course, one of the things that she tried to do was get a co-crystal structure with substrate, and she wasn't able to do that in the right time frame. But what she did do was she took our enzyme, and she kept it with nothing bound in the active site, and she measured what's called a circular dichrosome spectrum. And she saw this black curve. If she took the same enzyme and she bound it with an inhibitor in the active site, then she saw a change in this little lump. And for those of you who are not chemists, that little lump is really important, because that tells us that about 22% of the helix in our protein went away when we bound substrate. So that suggested to us that this orange structure Shravanti saw was present in solution, and when you bound substrate, it turned into the green structure and looked like all the other caspases. About that time, unfortunately, we ended up getting scooped by a Chinese group, Xiaodong Su, who solved the structure of caspase 6 with substrate in the active site. And this is their structure in blue. So if you compare our orange structure to this blue structure, you'll see the blue structure looks just like all the other caspases. And we know that this region can undergo a conformational change from the helix to the strand upon binding. But our circular dichrosome spectra showed us that it's in the orange conformation in solution. So we were really interested in this region I show you here, because it looks like it may be unique compared to all other caspases. And what we further noticed was that there's a cavity here that's just the size of a drug molecule that looks like we could maybe exploit it. And this helix, this cavity, can only form in the orange structure, but not in the blue structure. So that means, and it looks to us like perhaps caspase 6 is the only caspase that can make the orange structure. If that's true, Now we have a new allosteric site that only exists in caspase 6, but no other caspase, and that's exactly what we were looking for. So Shravanti made a series of mutations to probe whether any other caspase would be able to form this helical structure. And I'm summing up an entire paper in this one slide, but what she found was that no other caspase could form this helical structure, and it was precisely because of a relatively simple reason. So if you look at caspase 3 or caspase 7 or any other caspase, in that region they all have a glycine and a proline, or at least one of them. So 3 and 7 both have a glycine and a proline. A glycine and a proline are these special amino acids which are not good at forming alpha helices because they don't. Proline restricts the geometry of the helix, and glycine is missing a residue to make a hydrogen bond, so it destabilizes a helix. And if Shravanti took glycine or proline from either caspase 3 or caspase 7 and put them into caspase 6, now caspase 6 doesn't act like caspase 6 anymore. It acts like caspase 3 and 7. So what that told us is this orange structure is unique to caspase 6. And that was very exciting for us. It also turned out to be kind of lucrative for us because we found out that this helix, this extended helix and this cavity that I showed you are only present in caspase 6. So we were able to go to the Broad Institute and the NIH and form a collaboration to allow us to screen for inhibitors that bind at this allosteric site. And we ran the pilot screen about, I don't know, maybe about a year ago now, and the full screen of 370,000 compounds ran 2 weeks ago, and the screener then went on vacation without giving us the results. So I'm dying to know what came out of that screen of 370,000 compounds. But even in our pilot screen of 10,000 compounds, we already found compounds that inhibit caspase 6 with 600 nanomolar affinity and with about at least 1,000-fold selectivity over the most closely related caspases. So we're really excited about this project because we think we're going to find the kind of inhibitors that we want that are specific for caspase 6. And in the best case scenario, these could lead us to more interesting treatments for Alzheimer's disease. So that's work that's ongoing. So this is about the time when Ellie Velasquez, Edwin's friend, got involved in the project. And Ellie is really terrific. I don't know if you can see this picture, but he's one of the most unique graduate students I've ever come across because this is Ellie as a scientist, but before he came to UMass as a graduate student, he was actually a professional baseball player. He was a pitcher, and that's him holding his own baseball card. And I have a copy of that baseball card in my office. So anyone who wants to can come to UMass and see Ellie's card. But Ellie got the idea that, we found this new allosteric site in caspase 6, but maybe there's other allosteric sites which are equally exploitable. And nature has sort of given us a record of allosteric sites because of something called phosphorylation, which tells us about cryptic allosteric sites. And so Ellie said, if I want to understand phosphorylation, I can use a phosphomimetic version. So phosphorylation is putting a phosphate group onto a hydroxyl, and an amino acid that has a hydroxyl is serine. So he said, if I take caspase 6 and I make the mutation S257D, then that enzyme looks like it's been phosphorylated. If he expresses wild-type caspase 6, it cleaves to the two active bands, the large subunit and the small subunit. But if he makes that phosphomimetic version that looks like it's phosphorylated, the prodomain is cleaved off, but we get no cleavage between the large and small bands, which makes it look like the enzyme is inactive. And in fact, if we measure activity, it's totally inactive. He said, well, that's not surprising that that's inactive because we know that if caspases aren't cleaved, they're not active. So I'm going to make a new version of the gene where I can express the large subunit separately from the small subunit and then ask if the enzyme is active. So he did that, and even when the enzyme is fully cleaved, there's no activity. He sees a total loss of activity. And that is the same as when he takes caspase 6 and phosphorylates it with the known kinase, ARC5 kinase, that phosphorylates caspase 6. So he says, I've got a strong phosphomimetic. I want to understand how this phosphorylated enzyme gets inhibited. And so Ellie said, I'm sure all that happens is it gets phosphorylated, And that phosphorylation locks caspase 6 into the orange structure, the helical structure, and we know that's inactive because it can't bind substrate. And so he said, well, I'm going to look around at the residues that are near, here's the site that gets phosphorylated, serine 257. I'm going to look around and see if there's any charged residues that have a positive charge that could interact with a phosphorylated version of the enzyme. And so he did that. He looked around and he said, okay, I know the wild-type enzyme has this kind of catalytic rate enhancement. And if I make the S257D mutation, then I knock that down by 2.5 orders of magnitude. I bet if I mutate any of these positively charged residues to negatively charged residues, that if that mechanism is intact, then I should see that in the kinetics. So he made all of these positively charged residues into negatively charged or neutral residues, but he essentially saw no impact on the kinetics. These enzymes are all still dead. So he said that can't be the explanation. I'm going to have to solve the crystal structure. So he solved the crystal structure, and he was really surprised because his structure, which is in blue, looked nothing like the orange structure that he expected. So he said, that's a surprise, but I'm going to superimpose my blue structure with the structure of the active enzyme with substrate bound, solved by the Su group, and that's what we show here in red. And you can't see it at all on this slide, but don't worry, I'm going to zoom in. But what he saw was this particular loop which was pointing up in the active conformation, is pointing down in his structure. So I'm zooming in on that region of the protein, and here you see the L2' loop, which points up in the active structure, but in his structure, it's folded down. And the reason why it looks like it's folded down is because it looks like there's a steric clash between this phosphomimic and that loop. So he says, I wonder if that steric clash is important. If it's important, if I leave it the same size, I should get no change in activity, which is what he saw. If I make something that's not charged, but as big as aspartic acid, so this is an amino acid called asparagine, then I should see the same loss in activity, and that's what he saw. But if I make it bigger, activity should be dead. And it was even a little bit more dead when he made tryptophan. When he zoomed in a little farther, he said, actually, it looks like this S257D bumps into this particular amino acid in the L2' loop, proline 201. Well, it turns out that with molecular biology, you can do cool chemistry. You can shave off those 3 atoms, and you can ask what happens to activity. And so Ellie shaved off those 3 atoms. He changed proline 201 to glycine, and he said, this is what I predict the structure would look like. And he made that mutation, and he gained back 2 orders of magnitude in activity. So that was the home run, because that told us that the way phosphorylation inactivates this enzyme is by steric inhibition and a steric clash with this loop here. So this got us really excited about phosphorylation, because now we know about another allosteric site that's in caspase 6 only. And we also found that there's lots of allosteric sites, there are lots of sites of phosphorylation in all the caspases, and some of them, like this one in caspase 8, look like they function by a similar mechanism. So Ellie started to get, I didn't mean to leave this slide in. Ellie started to get excited about, I think I have another idea for how I could look for a new allosteric site, and it has to do with zinc binding. So Ellie found that zinc inhibits caspase 6, but no other metals inhibit caspase 6. And if you use a peptide substrate, the IC50 is 3 micromolar. But if you use a peptide substrate, then the IC50 is 0.37 micromolar. It's much, much stronger on a protein substrate than on a peptide substrate. So this was interesting to us because this said, well, maybe the zinc is binding somewhere that interacts with substrate, but outside the active site, and that would be very revolutionary to find an exosite for protein binding on a caspase. So Ellie came back to X6A, and with the help of Vivian and Jeanne and Edwin, he was able to collect a special kind of data set where it was tuned at the zinc edge. And so he collected this anomalous diffraction pattern and was able to visualize exactly where the zincs were in his structure. And when we zoom in on those zincs, we could see the 3 residues that are liganding the zinc. And one of the things that was really amazing about this was that one of the residues was a lysine. And traditional inorganic chemistry says, well, lysine should not be a good ligand for zinc. But Ellie looked around the PDB and found that about 3% of structural zincs in the PDB actually use lysine as a ligand. But he said, if I really want to understand whether this zinc binding site is an important contributor to how caspase 6 gets inhibited by zinc, I can test that explicitly. And so that's what Ellie did. So he made a series of mutations. If he took the wild-type enzyme, he saw that in solution, it bound 1 zinc per monomer. If he knocked out the catalytic cysteine, which could be a great place for zinc to bind, he still saw 1 zinc binding per monomer, so then he knew that this was not the binding site. We had discovered another allosteric site for zinc binding in caspase 9, and if he knocked out that site, he still saw 1 zinc binding. But if he knocked out any of these 3 ligands that were predicted by his crystal structure, then he saw no zinc binding. So that told us this was really the zinc binding site, and that this zinc binding site was what was leading to inhibition. And he also found that caspase 6 was unique because it was the only caspase that bound only 1 zinc. Oh, that's not true. Caspase 7 does too. But caspase 6 binds 1 zinc. Caspase 3 binds 3 zincs. Caspase 7 binds 1. And caspase 9 binds 2. And we've found the 2 sites where this caspase 9 binds. But the other cool thing about this result was that caspase 6 binds 1 zinc. We can see where that zinc is. We know it's not the active site. We know from crystallography exactly where it is. This is a second piece of information that tells us that caspase 6 is in the helical conformation in solution, because if it were in the active conformation, it would bind zinc at the active site. So this was really nice conformation. So then Ellie graduated, and he's now a postdoc at St. Jude's Hospital. So now we know that a lot of allosteric sites exist on caspases. Here are 4 that we know about on caspase 6. And we know that the constellation of allosteric sites are very unique to each protein, even highly related proteins within the same family, and therefore they might be interesting in drug design. In addition to being interesting in drug design, I have like 2 slides before I get on to the mentoring part, which I know many of you, that's what you came for. Let's see. So we wanted to take advantage of our understanding of these allosteric sites for another reason. And so this is the work of Witold. And Witold didn't get to come to the synchrotron until he was like a 4th-year graduate student. But I will never forget the day that we came here for the first time. And he walks into NSLS, and he looks around, and he says, whew, now I'm glad I pay my taxes. He was just so in awe, because this is our X-ray detector at home, and it's nothing like NSLS. And so Witold solved a bunch of interesting crystal structures, but I'll just tell you about one. He said, understanding that these loops move around and that their movement is critical for their ability of caspases to be active or inactive, I'm going to design a disulfide bond that locks them together. And it turns out this was a little more tricky than it seems. He had to engineer out some disulfides, and he had to try various things, but he finally found this version of the enzyme, which is a handcuff caspase. And so what's really cool about Witold's handcuff caspase is that it's inactive in any oxidizing condition. So extracellular solution is oxidizing. So what we've done now is we've attached caspases to polymeric delivery vehicles, and we've shown that we can deliver caspases to cells and kill cancer cells. But then once the caspase gets into the cell, it's released from the delivery vehicle. This disulfide is broken, the handcuffs come off, and that caspase becomes active and kills cells. So now we're using caspases as a drug themselves. So this is a killer protein, right? If we can get it into cancer cells, we can cause those cancer cells to die. So this is a fun project that's still ongoing. Okay, so I'm going to shift gears now from the science part of my life to I don't know, all the rest of my life and tell you a little bit about mentoring. And in my house, I don't know why, but for the last 3 years, we've gotten Sports Illustrated for free in the mail. And so every week, I read something called The Numbers. And so I want to play this game with you. So I'll give you a number, and then you can tell me what this number is. So 491. Any guesses?
Home runs?
Home runs. Okay, that's a good guess, but it's the number of faculty in my college in 2009. 94. Number of female faculty in my college in 2009. So it's not bad, but it's about 20%. 4.
Number of tenured female faculty.
Thank God that number is higher. But that was an excellent guess. It's the maximum number of female faculty in any one department in my college. And 2.25. We did. It's the number of research-active female faculty in my department, because there's 2 of us who are full-time and 1 person who's 25% in my department, 75% in biochemistry. So we looked across our university and we said, there's a lot of science and engineering women. There are 491 of us, but there's no more than 4 in any department, so women tend to feel like they're alone, even though there's 94 of us, which is a big number. And so this is my colleague, Lynn Marie Thompson. She's the other full person in my department in 2009, and that's my son when he was a baby. He's now 5. And Lynn Marie and I had the idea to apply for a mutual mentoring grant that was offered and tried to start this new initiative. And we got our idea from this book, which is called Every Other Thursday by this woman, Ellen Danielle. And what really caught my eye about this book was this person right here, because you might recognize her. This is Judy Klinman. And I also recognized Carol Gross. So they're both National Academy members. Liz Blackburn is also on there, also another National Academy member. And Judy Klinman was my professor at Berkeley when I was a graduate student who taught me enzyme kinetics. And this woman was amazing, right? And in this book, it talks about a mutual mentoring program that they started when they were assistant professors, before any of them were National Academy of Science members, that has proceeded on for, it was 20 years when this book was written, and they've now been going, and they've been going 30 years now. And so they describe how they run their mutual mentoring group, which was every other Thursday, they would get together at the houses of one of the group members, and they would work on their scientific careers. And so the book talks about how they ran their mentoring program and what are some of the obstacles that they faced. It also discusses the framework for how to set up one of these mutual mentoring groups, and it was just an inspirational book to us. So we said, we're going to send out emails to all the women faculty in departments that are 1 degree related, 1 degree separated from chemistry, which would be biology, biochemistry, polymer science, chemical engineering, and invite these women to a meeting so we can tell them about the book, and then ask if they'd like to be in a group like this. So we invited 40 women, and out of the 40 women, 18 women showed up at the dinner, and all 18 of them were interested in forming a mutual mentoring group. And we had assistant professors, associate professors, full professors at the meeting, and we really wanted to make groups that had women at all levels in each of the groups. And so we were trying to say, well, how should we divide? And I, at the time, had a little kid, and I wanted to have my mutual mentoring group at night when my kid was asleep. And there were other people who did not want to have it at night. They wanted to have it at lunchtime. And it turned out that exactly half of us had a strong preference one way or the other. So we didn't divide based on anything other than when you wanted to meet. So we have 1 lunch group and we have 1 night group, and we've been meeting since 2009. And so my group is every 3rd Wednesday, and we meet at somebody's house. In my group, we have 3 full professors, 3 associate professors, 3 assistant professors. It wasn't always that way. 1 associate professor decided after about 6 months it wasn't really for her. 2 of us got tenure, and we got a new assistant professor and a new full professor who's a distinguished, she has some very fancy title, distinguished professor of the university or something. We have 2 chemists, 2 biologists, 1 polymer scientist, 1 microbiologist, 1 kinesiologist, 1 veterinary scientist, and 1 biochemist. And I think our mutual mentoring group works, and my evidence is that people have been coming for 4 years, and everybody seems to value the mentoring group. And One thing that Peter and I were talking about earlier was that the mentoring isn't only the full professors telling the assistant professors how they should do it. Mentoring is working in all directions. And it's been really great. So after the first year, we had some money left on our mutual mentoring grant, and so we invited Carol Gross, who's an HHMI and National Academy member from the University of California at San Francisco, UCSF, to come and give a scientific talk and a talk about mentoring. So she called her talk Strategies for Success, and talk about her experiences with Every Other Tuesday. And so she came and she talked and she gave a fabulous talk. And then afterwards she wrote us this note, which really I thought was meaningful. She said, talking with all of you was one of the highlights of my life. I thought, oh, well, that's probably a bit of an exaggeration, but it was really nice for you to say. I had such a wonderful time and have told so many people what a spectacular support system you've built. Going around the table and hearing each of you talk was truly amazing, and the number of people who came to hear about mentoring was astonishing. It really meant a lot to me to visit with you. Thanks, and please share this belated thank you with all your wonderful group members who did so much to make my trip memorable. And we were so floored by this because we all thought her visit to us was really, a highlight of our life. And so for us, it was really useful for us to get to meet one-on-one with somebody who's been doing it for 30 years and who has really benefited from mentoring. So out of this group of 8 women, they started out as assistant professors doing this mutual mentoring. And they talk in the book about, Judy Klinman talks about how she spent a lot of time getting help from her group, preparing the perfect talk when she needed to overthrow the dominant paradigm about proton tunneling in proteins. And, they really helped her strategize how to cross this threshold. And with her help, her groundbreaking work was really well accepted. So for us, it was really, really inspirational. And so once Carol talked, probably 200 or 300 people came to hear her talk about mentoring. And after that, women in the College of Engineering decided they wanted to start their own mentoring initiative. So they now have a lunchtime group or 2 that meet. And then a couple years later, our graduate students who remembered this event a graduate student came to talk to me and said, I would like to start mutual mentoring because I feel alone. I'm the only Caucasian woman in my lab, and I feel like they're all wonderful lab mates, but they don't get me. And so one of the things that Lynn Marie and I got Lynn Marie to help me. We got a little money from the department, and we helped the graduate students have an organizational dinner. And they got together. We told them about how our system worked, and they decided they were interested, but they didn't set anything up that night. And as a professor, I really wanted to say, okay, now let's have everybody sign up on paper, and let's get groups, and who wants to meet now? But I didn't. I held back because I thought, I think mentoring works best when it comes from grassroots. That's how it felt good when I wanted to do mentoring. And, and my peers seemed really accepting to something that came organically from other people like them. And so I wanted to give the grad students the same opportunity. And if they grabbed hold of this idea, great. And if they didn't grab hold of it, fine. So it was really hard for me, but I walked out of the room not saying a word and thinking this was a total failure. They didn't organize anything. They're never going to get their act together. But by the next week, they had already organized 3 groups, and they were meeting, and their groups have been meeting for 2 years. And I had a student, Samantha, who was in one of those groups, and her group really helped her make some really hard decisions around being a postdoc, where to be a postdoc. And so I've seen that this model, not only did it work for these women, it's working great for my group, and I've seen that other people have found that it's a mechanism that doesn't take any money, right? If you're meeting in a conference room at lunch or you're meeting in somebody's house, you don't need money for mentoring. You don't need psychotherapy. You just need a supportive environment. And so this has really worked well for us. And so it's something to consider while you're thinking about how you want to structure your mentoring program. Of course, I'm a faculty at UMass Amherst. Next time you're in Amherst, come to visit. And I'll skip this. Most people are not biochemists. We're trying to get more people to publish in our journal because just this year, our impact factor passed JBC. So we were really excited about this on the editorial board of this journal. And so, but anyway, I'd like to thank the people in my lab. I really feel fortunate that I've worked with this amazing group of undergrads and graduate students. You can see sort of them growing up through the years. And I'll thank these groups that funded our work. And I really want to thank Vivian for the invitation. It's been really nice to be here. So thank you all. So that's actually, even though we're in different departments, we're all kind of in the same field. That's why we sort of chose the departments the way we did, because we thought those are the departments that sort of have the same tenure expectations and publishing expectations and the same kinds of journals. So that was one reason why we chose the group that we did. We wanted to have people from lots of different departments within a group, and the graduate students, when they organized, also wanted to have people from different departments rather than all chemists, because one of the benefits is that if something's not going well in your department, you can bounce that off your group, and they can say, oh, in our department it's even worse. Or they can say, in our department we do this and it really helps. And so just having a slightly different background is good. And so we did think about that a little. One interesting thing is in the book, there was a case where there were two people in the same department, and one was on the tenure committee of the other who didn't get tenure. And they talk about this in detail in the book, how that could have ripped their group apart, you know? But they had sort of some frameworks in place about what was okay to talk about and what wasn't okay to talk about. And so, like, I think there was one time when the person who didn't get tenure really needed to talk to the other people. And so that person who had been on the committee said, you know what, I want to not come because I want our group to stay together and function, but I want her to have a time to talk about those issues, you know? And so even though it was tough, they worked through it. And so I do think it was useful to read the book to sort of, because they had 20, by the time they wrote Ellen Danielle's the one that didn't get tenure. And then she went on to have this amazing, successful career afterwards. So it's kind of a nice story in that way. But I thought it was useful to hear about people who had been working under this model for 20 years and had therefore come up with some frameworks or guidelines for how to make this kind of mentoring work. Because if you are talking about professional things, and they are people who are at your same organization, there are, you know, like sometimes we try to say, well, my collaborator, and everyone in the room knows who the collaborator was. Like, we don't name names, but you don't have to because we know that person only has one, you know, collaborator or whatever. So, that is, it's a consideration, yeah.
Were there PhD level faculty members below the level of assistant professor who were not invited to join?
Yes. We only invited tenure. So at UMass, there's only lecturers or tenure system faculty, and we did not invite the lecturers, not because we don't like them, but they just have a completely different career path than us, right? And so we personally felt that because research is sort of so all-engulfing, and especially balancing research with teaching, we just thought, and we didn't include everyone. We didn't include the computer scientists either. We wanted it to be a little bit narrow so that we could feel, like, for example, my two best friends are humanities professors, and they are great, but they don't get what it's like to be a scientist. They really don't. My husband is a humanities professor, too. I don't really want them in my professional mentoring group because they'd be like, well, why don't you just go write in a cafe? And I'd be like, because I got to work with bacteria, and they don't want me bringing the bacteria into the cafe, you know? So that's why.
Very nice to hear about it. I'm just wondering, so how does it work exactly? Like, is that every week you have something in the agenda and then talk about a specific topic, or just like chatting, people sit around and share? Or is it like one speaker to lead or something?
Yeah, I can address that. So at the beginning, you know, we didn't know each other well at all. And so what we did for probably the first 4 or 5 meetings, at the beginning we used to meet every other week for the first while. And then we decided every third week worked better for us. But for the first 4 or 5 times that we met, we actually took a chapter from the book, and we talked about that broadly, because they were all things that related to us. You know, a lot of them were about time management, goal setting, how do you work on the most important thing. I mean, we really identified with the same issues that the women in the book were identifying with. But after sort of 4 or 5 meetings, we found that we didn't really need that framework anymore, because we were comfortable enough that we could talk about our own problems that were more specific to us. Those were problems that were general to everybody. And so the way it works is we go to someone's home, and whoever's home it is the host. And the host always speaks last. That's just a convention that the book set up and we adopted. And then we go around the room and we ask how many minutes they want. And we don't the host doesn't hold them to their number of minutes with a stopwatch, but we do kind of watch the time. So normally you're allowed to ask for 5, 10, or 15 minutes. If you ask for 45, then we know you have a big issue, you know, and that just basically means buckle your seatbelts. I have something, catastrophic to say. But mostly, like if it's my turn to go, then we go around, we say who wants to go first, and this is actually described in great detail in the book, and the person who is going states what it is that is the issue. I have this collaborator, this is an example I used earlier today, which really has come up in group. I have a collaborator, we have this paper, my student can't graduate or get a job until we publish this paper. Collaborator's been sitting on it for 6 months. How can I get them, I really want them to respond. And then usually the speaker asks a specific question, like, do you think it would be all right if I wrote to their wife and asked them to prod their husband to respond, to read this paper, And then we all can weigh in on that suggestion and maybe offer other suggestions. We can say, well, I had a collaborator that also sat on a paper for 6 months. What finally did it was I sent them flowers, or I sent them a babysitter, or whatever. So we sort of brainstorm around the topic, talking about if we have any ideas, but ultimately it has to come back to the speaker. And then the speaker has 2 options. They can say, yeah, I still don't know what to do. I'm going to keep thinking about this. Or what happens more of the time is that the speaker on the spot says, yeah, I think that's a really good idea. I think instead of writing them more emails, which are completely ineffective, I'm going to go speak to them in person. And even though I'm scared about it, I'm going to do it. And then we have anal people and not anal people. I'm on the anal side, so I have a little journal that I bring to group, and I then write down my goal, and I report to the group next time. And 2/3 of the group doesn't do that. There's about 3 of us that are really anal, and we write down our things. But I like to do that because it sort of keeps me focused. There's some accountability there that I'm going to go and tell. I want to be able to tell my group so that it motivates me to do the thing that's my goal. And so anyway, does that kind of explain? Yeah. Yeah.
No, there is some research that people who write down goals and action plans have a much higher percentage of success rate than people who don't. So anal or not, there's a benefit.
Okay. Good. All right, well, thank you guys for coming.